Very good afternoon to all. On behalf of Department of Science and Humanities, National Engineering College, I welcome you all for the 5th day session. I hope you learn something new on each and every day. So today's topic is mitochondrial biology, the fate of glucose. So once again, I welcome you all, and now I welcome Dr.Soni Deshwal. And now I request Dr. A. Anaraj to introduce our resource person. Thank you.
Okay. So thank you Ramachandran. So on behalf of our management and organizing committee of this 5-day, happy to welcome all of you for this 5th webinar on mitochondrial biology, the fat of glucose. Actually, we plan 5 talks. So we started in the Monday, and so up to Friday we have covered various topics from carbon cycle, astrophysics, and so many things we covered. So in this row, so today's topic is mitochondrial biology, the fat of glucose. Okay. So for delivering this lecture, we have with us Dr. Soni Deshwal. I have happily invite her for delivering this lecture. So brief introduction about Soni Deshwal. So actually, she completed BTech degree in Amity University in the year 2012. After that, he went to University of Leicester, UK, for doing Master Science in Molecular Genetics. After that, he moved to University of Padua, Italy, for doing doctoral research. So after his PhD, Humboldt. Okay, she's in Germany right now. So today the topic mitochondrial. Third, I welcome the resource person, speaker of the today's session, Dr. Soni Deshwal. So Doni, okay, thank you. Soni Deshwal for delivering the lecture. Soni Deshwal, you can deliver your lecture.
Okay, thank you very much. So first of all, hello everyone, good afternoon. It's still morning in Cologne. I would first like to thank Dr. Ramachandra for inviting me to give me this platform to present to you some of the biological things that I have learned during my PhD and postdoctorate and also before. And I'm very happy to deliver this lecture today. So can you all hear me properly?
Yes, we hear you.
Okay, perfect. So I would just share my screen now, and so I just want to say that I prepared a conceptual lecture more to basically have like a bit of brainstorming to see how the field is and where the field is going. So please feel free to ask me any questions in the middle or at the end, how you would like it. All right. Can you all see my screen now?
Yeah, it is visible now.
Okay, perfect. So, so first of all, thank you very much for inviting me to the talk. I'm really glad to present. It's my first time actually presenting in an Indian university, so I'm really excited. Okay, so this is the picture that we actually take from the microscope of a cell. And I know that it's very, very different than what we are actually used to see in the So it's actually very, very different what we are used to see in our textbook when we learn how actually the cell looks like. So this is a nucleus in the middle that you can see in this blue color, and these red spaghetti-like structures that you see here are actually the mitochondria. And as you can see, that it's not that the mitochondria is a single organelle and staying itself, but it is actually forming a very nice network with other mitochondria as well. So in this red color, whatever you see here are actually all the networks that you see are formed by the mitochondria. So this is how actually a cell looks like if you look into the microscope. And right now you're just looking at the nucleus and the mitochondria here. So I wanted to start with basically how the mitochondria ended up being in the cell. So this was not always the case, but billions of years ago, what happened was in a cell, a bacteria, a photosynthetic bacteria, entered into the cell, and actually these bacteria are the ones that then became mitochondria over the time. So basically, it's more like a relationship between a host and a parasite, but more in a healthy way. So I have to say that the difference between the prokaryotes and eukaryotes is actually this. And the fact that mitochondria or this bacteria entered into the cell, it gave the cell a lot of advantages in many different ways that we are going to learn today. So also from the textbook knowledge, one can imagine that there is one mitochondria into the cell, but that is actually not the case. A cell contains multiple copies of mitochondria. And a mature egg cell, that is where we have the highest number of mitochondria per cell. It can actually range from 100,000 copies of mitochondria to 600,000 copies of mitochondria, because this is the mature egg cell is the cell that needs a lot of energy, of course, to do all the work that it has to do. So this is how the bacteria ended up into the cell and what we call to the mitochondria. So one of the most important features of mitochondria is actually its structure. So I wanted to talk to you a bit more in depth about the structure of mitochondria, because it's very, very important, and scientists have put a lot of effort to actually understand how this organelle is actually in the 3D. So I think you might have all learned into your school in 10th grade that there is mitochondria, which has an outer membrane and an inner membrane. So what is actually the function of these membranes? So the outer membrane is rather more porous, which means that the metabolites can actually easily enter from this membrane inside the mitochondria. And the inner membrane is the one which is more impermeable, which means that this is basically a very strong protecting layer for the mitochondria that protects it in its own environment. And of course, now this mitochondria sits into the cell. But if you imagine billions of years ago, these bacteria were exposed into the environment and they had to protect themselves. So anything that enters into the mitochondria or that goes out of the mitochondria is very, very tightly regulated. And the health of these organelles are very, very important for a cell to maintain its homeostasis. So the inner membrane is the one that is more impermeable. And inside, then this blue color that you see here is actually the matrix. And this is more the liquid part, or the part where you have all the soluble things. So anything that ends up in the matrix has to be soluble. And this inner membrane is basically the border that keeps it separated from the outer membrane and the matrix. So there are different features of the inner membrane and the outer membrane. And one of the very important feature is that the outer membrane is made up of more phospholipids. And these are the lipids that we get from the food that we eat. And the inner membrane is made up of both proteins and phospholipids. and the ratio of proteins to phospholipids is very, very higher in the inner membrane. So that means that this inner membrane is basically the house for several proteins inside the mitochondria. And not only this, but as more and more research is progressing, we are actually learning that this inner membrane is very important for several activities that are happening inside the mitochondria. The area of the inner membrane is much more than the outer membrane. As you can imagine, that this inner membrane is basically folded in several folds, and this gives this inner membrane the flexibility to have much more area than the outer membrane. And when the inner membrane is folding, it creates these junctions which are called or known as cristae. So when we say cristae, we actually means these folds that inner membrane is actually creating. So before it was thought that the inner membrane is really large and it is folded because it needs to host or provide the house for lots of proteins to stay in, right? But now more and more research is telling us that these cristae are actually not only just the junctions, but there are several metabolic activities that are actually happening in these cristae. And that is very interesting to understand. And also it has been shown now that the organs where you have a very high demand of energy, for instance, heart or skeletal muscle, if you imagine that your heart is beating from the day that you are born until the day that you die, that means that heart really requires a lot of energy that has to be made into the mitochondria. It's very important that these organelle maintains these cristae junctions. And if these junctions are not maintained, that is one of the pathological signs basically into the disease. So moving ahead from this, another interesting fact about the mitochondria is that it has its own genome. So I think you all are aware of it, that we have a nuclear genome which we basically inherit from our mother and father. The same way we have a mitochondria has its own genome. So what does that mean? That means that over the evolution, this bacteria that entered into the cell basically kept its own genome, and that's why the mitochondria now has its own genome. But just to give you roughly a comparison, a human genome contains around 6.5 billion base pairs of the nucleotides, whereas a human mitochondria contains only 16,000 base pairs. So you can imagine that how tiny this genome of mitochondria is as compared to the nuclear genome. What does that mean? So why do we need a DNA in general? So we need a DNA so we can encode for a specific gene which can encode for a specific protein. And these proteins are the enzymes that we need for any function that we have to do inside our body. So it's very important. But that means that mitochondria can only encode for roughly 13 proteins. So 1-3, only 13 proteins, which is very surprising because a mitochondria is the house for more than 1,000 proteins. So so this was a very major finding in this field when people realized that mitochondria can only encode for 13 proteins, and the rest of the other proteins are actually coming from the cytosol inside the mitochondria. And this tells us that how important the communication of this organelle is with the rest of the organelles inside the cells. So the harmony should be very, very well maintained between mitochondria and other organelles, because mitochondria also rely on the fitness of other organelles inside the cell to basically work very properly. How these proteins that are synthesized in the cytosol can enter into the mitochondria, we'll discuss it later in my talk. Now, where is this mitochondrial DNA actually coming from? Have you guys ever thought about it? And the answer is that this mitochondrial genome is actually coming from our mothers. And the very peculiar thing about this mitochondrial genome is that we only can inherit this genome from our mothers. This also shows us the supremacy of the woman actually into the nature, and how evolutionary the male mitochondria has basically will completely disappear. And the only mitochondria that is going further and further in our next generations is actually the mitochondria from the woman. And I find it really fascinating. How does it actually happen then? So we know that woman actually gives the egg, so the ovum, to the child when the fertilization is happening, and the male donates the sperm. And there is then the mixture of ovum with the sperm, which gives a rise to the egg, where basically which basically develops into an embryo. And these sperm actually penetrates the ovum only with the head. So what does that mean? So this sperm actually, if you look here, so this is the ovum here. It has a nucleus, this black thing, and it has these tiny mitochondria. The same way, this is the sperm here, and it has the nucleus on the tip of the head, and then it has a mitochondria in the rest of its body. When the sperm penetrates the ovum, it actually penetrates it only from its head, which means that it is able to donate the nucleus to the egg, but it is never able to donate the mitochondria to the cell. So what is happening here is that the sperm that is left over after penetrating the nucleus, the sperm still contains the mitochondria, and it is basically discarded by our body, or by the body of the woman. So at the end, what we get is we get the two nucleus, which is from mother and father. That's why we have the DNA that comes from mother and father both, but then we have this mitochondria that is actually only coming from our mother, which also means, by the way, that if your mother carries the mutations into the mitochondria, this will be, of course, also transferred to you. And that can also be, of course, become a problem, because usually the genes are always naturally selected for well-fitting of the good survival of the human being. But in this case, if the mitochondria is defective, we cannot take the advantage of the mitochondria from our father, because we would only get it from our mother. And now there are lots of studies going on into the in vitro fertilization field, where they are trying to put the mitochondria from another female into the egg to maintain the fitness of the egg if the woman contains the mutations. So this was about our lovely moms. Now going ahead about mitochondria, one of the feature of mitochondria is mitochondrial dynamics. So what does the term mitochondrial dynamics means? So first of all, the mitochondria can never be de novo synthesized, which means that we cannot produce mitochondria from zero. So we cannot synthesize a new mitochondria. That means that we have to work with what we have. So it means the mitochondria has to be constantly divide and constantly fuse. So there is a fission and fusion that is happening in the mitochondria at a regular basis, constantly. And this is what is called mitochondrial dynamics. So how is it actually happening is a very, very elegant process, and basically a mitochondria can be divided with the help of a protein called DRP1. And I don't want to go in detail here, but just to give you an overview, that the DRP1 is a protein that is recently discovered. So it's a very recent discovery that the DRP1 protein forms basically like a chain around the mitochondria, and it oligomerizes, meaning that it forms complexes with other proteins, other DRP1s, and it causes this constriction into the mitochondria. And then what happens is that this mitochondria basically divides into two. And like this, we have then two mitochondria from one mitochondria. At the same time, the fusion is happening, which means the merging of two mitochondria. And there, one of the most important proteins that is involved is actually called mitofusin. As you can guess from the name, it's mitofusin. So fusin means it's causing fusion in the mitochondria. And how that happens is that these proteins basically stay at the outer membrane of the mitochondria, and it tries to bring closer two mitochondria so that they can fuse, and the outer membrane then is fused. And after that, the inner membrane, there are other proteins that are sitting at the inner membranes like OPA1 and phospholipids that causes the fusion of the inside of the mitochondria. And this is how the inner membrane is actually fused, and you have a fusion happening. So this is a very important process that is continuously happening in the mitochondria. There is continuously dividing and fusion that is taking place. And before, it was thought that these one of the processes is important, so you need fission and you also need fusion. But now the research has showed us that actually the balance between these two processes of division and fusion is actually very, very important. And if you shift the balance towards one of the sides, so if you divide the mitochondria too much, or if you fuse the mitochondria too much, then it is not healthy for the mitochondria. Now, the question is that what happens if the mitochondria is not able to fuse, or what happens if this mitochondria is dividing too much or is not able to divide, or what happens if the mitochondria is not healthy? Then there is a way in the cell where they can get rid of the defective mitochondria. And this term is called mitophagy. So this is basically the death of the mitochondria. And this term is coined from a term called autophagy, which is coined by Professor Yoshinori Oshumi from Belgium. And he won the Nobel Prize in 2016 for actually coining this term called autophagy. And auto means self and phagy means death. So it means the self-death. So a cell kills itself when it is not healthy anymore. And this is also happening in the case of mitochondria, which we call mitophagy. And what happens here is that when a part of the mitochondria, or any mitochondria that is defective, then it can happen that this mitochondria is eliminated by the cell, and this is called mitophagy. So there also the process of division actually comes in very handy for the mitochondria, because mitochondria doesn't want to get rid of the whole mitochondria all the time. It can happen that only a part of mitochondria is defective. So what mitochondria can do is it can basically eliminate this part by going through division. So it can just divide itself and creates this damaged mitochondria like separately. And this damaged mitochondria then is very elegantly separated from the rest of the mitochondria and basically can go to death. Now, as I mentioned before, that mitochondrial communication is very, very important with other organelles. Is the same like how we humans live in our neighborhood, where the communication between our neighbors become very important, and how we rely on the society and how the society relies back on us. And it's more like a vice versa process. You need the society and the society needs you. And it's the same way how what is happening in these tiny organelles' life, that they also need to rely on other organelles inside the cell. So the communication between these organelles inside the cells is a very important and very intriguing field. And there is a lot of research now going on how these mitochondria are actually communicating with other organelles. And this is not yet fully understood. So the knowledge that we have today about how these organelles are communicating with other organelles is very, very limited, actually. And what you can see here is that there is this mitochondria that is in close proximity with another organelle, that is endoplasmic reticulum. So we have all learned a bit about endoplasmic reticulum in our schools. And endoplasmic reticulum, you can imagine this organelle basically is like a factory that is producing lots of proteins inside it. So it produces lots of proteins inside it, and then these proteins are localized to different organelles inside the cell. So it's just like a warehouse of Amazon, where it puts all the stuff that the city needs, and then it calls the delivery people, and these people basically transfer the stuff from one Amazon warehouse to your place. Or how you do online grocery, and there is a supermarket that actually contains all the food in the neighbor, and then someone can go there and pick up the food and then come back. And this is what is actually also happening with the endoplasmic reticulum, that it contains all the proteins inside the cell, and then these proteins are actually targeted to different places into into the cell wherever they are needed. So what are endoplasmic reticulum and mitochondria sharing between them? So not only the proteins, but also different metabolites they are sharing. And one of the important metabolites that they share between them is calcium. And I'm sure that you all know about calcium. You have it in your food, you eat it. We learned that calcium is important for your bones, calcium is important for our teeth, and calcium is important for our body to maintain. It's very important, and with age, we need more and more calcium to have like our bones very well. And in the old people, why the bones can break easily is because of this, that the calcium in the bones can go low. And that's why when an old person fall down or when a young person fall down, there is a huge difference between it. A young person maybe is able to take it, but an old person is not able to cope up with this falling, because the bones would just break because there is less calcium. So just to say that calcium is a very important metabolite in our body, and this calcium is actually transferred from endoplasmic reticulum to the mitochondria. And mitochondria does a lot of stuff with the calcium. It is important for the proper functioning of several enzymes that are located into the mitochondria. So why doesn't mitochondria have calcium to start with? That is because too much of calcium is also bad for mitochondria. And since mitochondria is one of the most important organelle in the cell, because it's the powerhouse of our cell, it's where the energy is produced, it is very important that the calcium entry into this organelle is actually heavily regulated. And this is done through endoplasmic reticulum. So there is a specific transporter that has to be present at the outer and inner membrane of mitochondria called VDAC and MCU, which is a mitochondrial calcium uniporter. And these transporters have to be present to pass the calcium from the endoplasmic reticulum to inside the mitochondria, so that mitochondria can generate ATP, which is basically the energy produced. So I would use this term ATP quite a lot, and this is basically what's the energy that is produced by mitochondria and then is used by all our body to do any function that we have to do, to get up in the morning, to metabolize our food, to go for a walk, to work, to sit, to talk, and everything that we basically do. What we need is the ATP. So to say here that why do we need these transporters on the mitochondria is because, as I mentioned, that the inner membrane of mitochondria is very, very impermeable. So you cannot just pass anything through this membrane. It has to be very, very specifically regulated. And then it is regulated by these transporters. So you can imagine these transporters basically like a gated channel. So it's like a gate. So it has to open, and when it opens, the calcium can go in, and there is another transporter which has to open, and the calcium can go out. So what happens if this gate remains open? If the gate remains open, then more and more calcium can just go in, and at some point mitochondria will swallow, because it cannot keep all the calcium inside, and it becomes too much for the mitochondria, and the mitochondria would basically just die. So it's very important for the mitochondria to regulate the entry and exit of this metabolite from the mitochondria. Okay. Another interesting thing here that I wanted to mention is that these two organelles are so much connected that if there is any kind of stress that is there on endoplasmic reticulum. And what do I mean by stress? There can be different stresses. It can be that that the endoplasmic reticulum has to synthesize a lot of proteins, and then it is not efficient in its job, which can cause a lot of stress on the endoplasmic reticulum. The same way how a factory can work. If you ask the factory to produce a lot of products, it can happen that this factory collapse at some point because the people are not able to work efficiently just because they are working much more than they should. And at some point, the people would just collapse. And is the similar thing that is happening with the endoplasmic reticulum, that it can collapse at some point. when a situation like this happens, then also first ER and mitochondria try to communicate and solve this problem. But if this problem is not solved and ER still remains under stress, then also it happens that at the situations of late ER stress, the mitochondria actually collapse. And this relationship between them is also vice versa. So if the mitochondria is not fit, it can cause stress, and the same way it can transfer this stress to endoplasmic reticulum, and also the endoplasmic reticulum can collapse. So once again, all this saying is to emphasize that how important this communication between these organelles is. And this was not understood from very, very recently. And only recently people started to understand that how this communication is so much more than what we thought previously. Another important aspect of mitochondria is actually its metabolism, and this is where the magic happens. This is where the mitochondria is actually generating energy. And mitochondria are known for the hub of metabolism of the cell. And the powerhouse of the cell definition is now considered very old. And this is now the new term, metabolic hub of the cell. These are mitochondria considered. So I guess in your carbon cycle lecture, you might have heard about how glucose is actually entering into the cell from your food, and how glycolysis is happening, and how glucose is converted into pyruvate. So glucose is entering into the cell from the food that we eat, and then it is going and converting into the pyruvate. And once it converts into the pyruvate, then it actually generates 2 molecules of ATP. And this is the energy that you need in your cell to function. So glycolysis, the process where glucose is converted into pyruvate, generates...
One doubt.
Sorry? Is there any question? Okay.
No. You can.
All right. So the ATP, only 2 ATP molecules are generated from the glucose to the conversion of pyruvic acid, and then this pyruvic acid is actually what enters into the mitochondria. So this glycolysis is happening into the cytosol, and then the pyruvic enters into the mitochondria, and then how pyruvic is converted into acetyl-CoA and how the Krebs cycle happens is another way how cells can actually produce energy. But once again, also from this Krebs cycle, what we get is only 2 molecules of ATP. So if you think about it, that how much energy we need, only these 4 molecules of ATP coming from the breaking down of glucose and Krebs cycle is actually not at all enough. So we need something more, and this something more is basically the place where mitochondria comes in picture. And to produce or to increase this production of ATP very, very high, or to push it, what happens is that the metabolites that are coming from these reactions like glycolysis and Krebs cycle, these are all given to the electron transport chain into the mitochondria, and this is where we can generate 34 molecules of ATP. So if you just now compare how much ATP we get from the breakdown of glucose and how much ATP we get from mitochondria, that is 34 molecules from electron transport chain and 2 molecules from Krebs cycle, which is in total 36 molecules. This is the energy that we are generating in the mitochondria. So it's basically 18 times more than the glycolysis. And here, one interesting fact is that the organs that demands most energy, for instance, heart or your muscle or brain, again, heart beats all the time, so it needs energy to spend. It has to spend energy to actually be able to beat. The skeletal muscle, whenever you're moving or your muscles are moving, you need to, we call it like you burn energy, right? And the energy that you're burning here is actually these ATP. And your brain, which is constantly functioning, even when you sleep, a human brain never actually rests, and it's always constantly working. So what these organs need is a very high demand of ATP. And these are the organs where mitochondria become one of the most important organelle, or the most important organelle in these organelles. It's also now more and more appreciated that if there is any damage done to the mitochondria, it will cause several problems into these organs. So if your mitochondria is not fit, then your heart or your muscle or your brain cannot be fit. So this is what we are now understanding. And there are several treatment strategies that scientists are now focusing on, basically treating mitochondria, because before it was not clear whether the mitochondrial dysfunction is the cause or the consequence of the disease. But now we understand it very, very well that mitochondrial dysfunction is actually cause for several disease. For instance, if you have a diabetic cardiomyopathy, or if your heart just fails, or if your muscles are not good, or if you have a neurodegeneration disease, or if you have Parkinson's, Alzheimer's, in all these major pathologies, actually mitochondria is become very, very important to function very, very well. And why is it like this? It's because these organs need so much energy, and therefore they rely more and more on the mitochondria. So we are going to now talk a bit more about how this mitochondria is actually producing ATP. So what is actually happening there? So inside the mitochondria, between the inner membrane and the outer membrane, there is a space that we call intermembrane space. Okay, so intermembrane space, the space between the two membranes, easy to understand. And what is happening there is an electrochemical gradient that is established in these organelles, and this electrochemical gradient is basically the basis for the production of the ATP that mitochondria has to produce. And how is it happening is once again our knowledge in how this process precisely happening is has advanced a lot in the last 10 years, but still very, very limited. So I'll try my best to explain this here. So basically the hydrogen ions or the proton are basically pumped out from the matrix of the mitochondria to the intermembrane space. And this is done by a specific chain, which we are going to talk about in the next slide. And when these hydrogen ions or when these protons are basically pumped into the intermembrane space, it causes this electrochemical gradient to form, because there is more positive charge into the intermembrane space as comparison to the matrix. So you have to imagine it in a way that that the intermembrane space is more positively charged field, and the matrix is more as comparatively more negatively charged field. And these hydrogen pumps are constantly pumped out into the intermembrane space. And this is the reaction that is happening in your body, in your cell, in each mitochondria every single minute or every single second. This is what is happening inside your body, and this is the source of the energy, how mitochondria is producing energy. So let's go more deep into the system and let's understand how exactly this is happening. So as I said, that this is happening via this chain, which is called electron transport chain. As you can understand from the name, electron transport chain. So it's a chain that is actually transporting the electrons from one complex to another. So how does this machinery looks like? So this in blue, you can imagine in the background is the inner membrane of the mitochondria. This space here, white, is the matrix of the mitochondria. And this space here is the intermembrane space, okay? And what this electron transport chain has is 5 different complexes into the chain. This is complex 1, 2, 3, 4, and 5. Okay, and this Q is basically part of the complex 1. So these are the 5 different complexes. How these complexes look like, what are the structures of these complexes? We know there's only about 2 complexes, so complex 1 and complex 5. The rest complexes are still a mystery, and crystallographers are trying to solve this structure with the crystal structure of these complexes, but so far no success. And what is happening now is that the electrons are passing from one complex to another. And in this process, when electrons are passing from one complex to another, the hydrogen or the protons are basically pumped out into the intermembrane space, causing this electrochemical gradient. So here, NADH, which we get from the Krebs cycle in the matrix, is basically converted to NAD+, and this hydrogen is then pumped out from the complex 1. And then these electrons that we get from here are passing to the different complexes. So these electrons then are transferred to coenzyme Q, then is transferred to cytochrome B, cytochrome C, another complex, another complex, another complex, until the end, where finally the oxygen is the molecule that is the acceptor of all these electrons in this process. So oxygen accepts all these electrons and gets converted into water, and which is H2O, and this is where this reaction actually stops. So when oxygen converting into the water is what we call that the reaction has been successful. So now you are able to, the transport chain work well, all the electrons are transferred to oxygen, is converted into water, and the hydrogen pumps are pushed into the intermembrane space. But still, how the energy is actually produced, we are just transferring here electrons, right? So to go into this, so I want to talk more in detail about this complex 1, and this is the complex that you see here in blue, and this is now the crystal structure of the complex 1. So this is how your complex looks basically in 3D. So this is the biggest complex of the electron transport chain, and the first one, a very important one. It is into this inverted L shape, you can imagine. And it is not only, it is basically submerged into the inner membrane and matrix both. Therefore, it is basically in contact with the mitochondrial matrix, and it is contact with the inner membrane. And what is this complex? What does this complex contains? So this complex has basically 44 proteins, and these 44 proteins, as I said, that mitochondria can only make 13 proteins. That means that this complex is made up of some of the proteins that are encoded by mitochondria and some of the proteins that are actually encoded by the nucleus. And that's how we get here 44 mitochondrial proteins. And what's happening here is that the electrons that we get from NADH are actually transferring to one of the subunit of this complex, and there is a chain of then reactions where the electrons are finally going to coenzyme Q, and it's converting into QH2, and then these hydrogens are pumped out one by one on each single reaction when the electrons are transferring. How exactly this reaction is happening, so how exactly the electrons are moving from one to another, and how hydrogens are pumping out into the intermembrane space is not really understood. What we only know is that it is coupled. So when the hydrogens are pumped out to the IMS, there is generation of ATP. So this is what we know, and this reaction is very well coupled. But how exactly this happens, we are not able to understood yet. And there are several labs now that are focusing on precisely understanding how this reaction is actually happening. Now moving on to the another important complex of this electron transport chain, which is complex 5, and this is the last complex of the electron transport chain. So this is this purple one or magenta one that you see here. This is the last complex, and this is where ATP is actually produced. And in fact, the name of this complex is ATP Synthase Complex, which means that the ATP is synthesized here. So this is again the crystal structure of ATP synthase. So this is how exactly it looks like in reality, and not this cartoon that I showed you here. So this is how exactly it looks like, and this structure has been solved by electron microscopy and crystallographer scientists basically. So what is this complex doing? So from complex 1 to complex 4, they are all focused in pumping out the hydrogen. The complex 5 does the opposite. It tries to bring the hydrogen back into the matrix, because it realizes that the electron chemical gradient is established, and now you need some hydrogens to pump into the matrix. So this complex does opposite of all these complexes, and that's where the magic actually happens. And how it happens is that now here it's inverted. So this is intermembrane space, and this is the matrix. So intermembrane space is the space where you have so many hydrogens, and matrix is where you lack hydrogens. So what this ATP synthase is doing, it tries to bring these hydrogen back into the matrix. And this is now a rotor rod, this in brown color. What happens is that hydrogen ion binds here, and when they all start to bind here, this rotor rod is actually moving 360 degrees. And when it is moving, the central stalk of this complex is also moving, and then the hydrogen ions are transferred to this F1, it's called F1 complex, and it's transferred to this F1 complex where hydrogen are finally reaching to the matrix. But what's happening here is that this rotor is again here, the movement into this small rotor into the inner membrane leads to the movement in the central stalk, which then eventually leads to the movement of this headstock. And when this rotor is moving into 360 degrees, this is where actually ATP is produced. So this energy, when it's moving, is something like you can imagine like a turbine. When the water falls from the dam and the turbine is moving, the water is actually constantly falling. Turbine is moving, producing energy. Is something similar what is happening here. These hydrogen are constantly falling on it, and this rotor is moving. There is conformational changes happening, and then ADP is converted into ADP. And bingo, we have our energy produced right here at this complex at this level. And this is also very, very elegant process, and it's very beautiful to see. You can actually see how this complex is moving with the 3D images and all, and it with the tomography, and it looks really, really fascinating. And so that means that the function of this complex is actually very important, because of course, if you keep transferring the electron, but if this complex is not working, you would not have any ATP produced. But this is not only true for this complex, it's also true for all the complexes. So whenever any of the complex basically stop working, the electron transport chain can collapse. And this is when, this is the time when there is no turning back, which means that once the electron transport chain fails, there is no rescue for the mitochondria. So mitochondria just go away, your cell cannot produce any energy, and eventually the cell will die. And if this happens in an organ like heart, for example, so it's very important that you all remember that in the heart, the cells are never dividing, which means that when you are born, if you have 10 cells in your heart, for all your life, you will have only 10 cells in your heart, which means that if you lose one of the cells in the heart, it's never recovering back. So you would never have it back. And this is when heart failure happens, when too many cells died in the heart, and heart cannot work properly because it needs a force of 10 cells, but these cells are dying and 2 of them is died, is only 80% of the heart that is working. And if eventually it's not enough to produce the energy that your body needs, your heart will fail. And this is one of the leading cause for death happening around the world. And this all starts at this level, so at the level of electron transport chain. And this is why it is very important that scientists understand that how we can prevent the collapsing of this chain. Because if this chain is not working, as I said, there is no returning point from there, and there is no rescue that can actually happen. And this is the reason that most of the mitochondrial labs are actually focused onto this electron transport chain. And now we have done a lot of development into the field, but still there are so many things that we do not understand. So one of the side effects of this electron transport chain is that it actually can also produce reactive oxygen species. And this is where I focused my PhD on. So I was really interested there to understand what is the role of reactive oxygen species that is produced by electron transport chain. So what does it exactly mean? It means that when electrons are transferred from one complex to another, the last source for the electrons is oxygen, right? And the oxygen accepts all the electrons and gets converted into the water. If the reaction stays like this, it's a perfect reaction. But sometimes it happens that these electrons don't go through all the machinery, but rather escape. So these are these naughty students into the class that start joining the class, but before they could finish the lecture, they just escape from the lecture, right? So you can imagine these electrons like those naughty students. And what is happening there is that these electrons, when they pass immediately from one of the complex, they can actually donate this electron directly to the oxygen, and then oxygen can become super oxide, which means that it's able to now more react because it has this one electron. When this superoxide accepts another electron, it becomes hydrogen peroxide. And when this hydrogen peroxide accepts another electron, it becomes water. So this is our safe thing, water. What we need is water. We don't need superoxide. Why don't we need superoxide? Because of course, if you have more and more superoxide, it's so reactive as a species. And this is where the name comes from, ROS, reactive oxygen species. So these are so reactive, these oxygen species, that they start randomly reacting with whatever they find in the place where they're generated. It means if they meet a protein, they will start oxidizing protein. If they meet a membrane, they will start oxidizing lipids. If they meet even DNA, they will start damaging the DNA. Once again, how this react? So these reactive oxygen species are not only bad but also good, because we know that these species you need for signaling, because they are so reactive, they're so fast that you can really use them as a messenger. So that's what cell does. Cell sometimes use it as a messenger to send different signals. Okay, so these are basically, you can imagine the guys with Ferrari that go like really fast and can reach immediately to some place to send a signal. If you want to send a signal, you want to use these guys because they are the fastest one. But at the same time, how they actually signal, again, is another very mysterious field that nobody really knows how they're actually signaling. But it's very important for the mitochondria to maintain the level of these species. So you don't want to have too much of reactive oxygen species, and in the same way, you don't want to have too less of reactive oxygen species. So too much of it is bad and too less of it is bad. So it's one of these complicated processes in the cell that is happening. And it's something similar what you can imagine with the sugar. So we need sugar in our body to basically do our daily activities. But at the same time, if you take too much of sugar, it can cause obesity. You can be diabetic. So that is bad. It's the same thing with the oxygen species. But mitochondria is a very complicated and very good into self-defending itself. So it's not so easy that you just generate these reactive oxygen species and they're gonna go around and keep damaging things. So it has a very good defense mechanism. And then when this happens, that these oxygen species are generated, which can cause protein oxidation or lipid peroxidation or mitochondrial damage, that's where the defense mechanism of mitochondria comes into picture. And just like every country has their defense mechanism, mitochondria also likes to guard themselves, and they have a very good quality control, which we call mitochondrial quality control. And this is where I'm actually now doing my postdoctorate in, and I'm very much interested in how this mitochondria is actually defending itself. So I change a bit from my PhD to postdoc to focus now into this part. And what is here happening is that we have different guardians. So when you want to have a defense mechanism, what do you need? You need people that can defend it, right? The same way mitochondria need different proteins that can be used as an army for mitochondria. And these proteins are called proteases. So mitochondrial proteases, these are very important protein in the mitochondria that have to work continuously to make sure that mitochondria is healthy. And that is the whole point. And what is happening here is that whenever reactive oxygen species damage proteins, these mitochondrial protease can actually chop off this protein and cuts it into small pieces and sends it back to the cell to use then to produce something different or to produce like another protein from it. So they recycle these things. So these proteases are basically very important, and they are residing into the different compartments of the mitochondria. So you have mitochondrial proteases in the outer membrane of mitochondria, in the intermembrane space of mitochondria, in the inner membrane of mitochondria, and in the matrix of mitochondria. And these green guys that you see here are actually mitochondrial proteases. So just for the simplicity here, I showed only 5 of them, but actually in total, mitochondria has 45 proteases. So there are 45 people in the army in a mitochondria that is actually taking care of doing these jobs. So I don't want to go here more in detail, but this is just the process to really say that why do you need proteases in different compartments of mitochondria is because a protein can be misfolded into the matrix, can be misfolded into the inner membrane, can be damaged into the intermembrane space, or can be damaged into the outer membrane space. So therefore, you need these guardians everywhere, because it's not so easy for proteins to move from one place to another, especially into the inner membrane, because it's very hydrophobic. So that's why you put these proteases in different locations, and you tell them that you have to now guard this location into the mitochondria. And I have to say that they do their job pretty good. And it's very rare that if you have a healthy mitochondria, that this defense system is not working. Usually, when we grow old, when we are becoming old, these are the proteins that then start a bit losing their function. And it has been now associated with many diseases that these proteases are not working properly when basically you are old, which actually leads to the accumulation of damaged proteins into the mitochondria. And as anything that is damaged, if you don't want to keep it in your house because it causes troubles, right? If you have too much garbage in your house, you want to get rid of it because it's not good, because it can be a source of infection. It can be a source of pathogens coming into your house, so it's not good. The same way the cells look at it in this way. If mitochondria has too much damaged protein, is not good, so they want to just get rid of it. And if you cannot get rid of it, it's a trouble for the mitochondria. So now I want to just focus a bit very shortly onto the last part of my talk, which is basically the mitochondrial protein import. So what is happening here? So why we want to talk about mitochondrial protein import? Because it is very important, because as I said, mitochondria can only synthesize 13 proteins, which means that other proteins have to come from outside. So if someone has to come from outside, there has to be proper channels how these proteins can actually enter. In this way, we can define that the protein in the mitochondria can be localized into 3 places. It can be in the matrix, it can be into the inner membrane, and it can be into the outer membrane. And it can also be into the intermembrane space. We can call this the 4th location. So how this is actually happening? We have to remember now, again, this electrochemical gradient is very important. So we have more and more positive charge into the intermembrane space and less into the matrix. So what does that mean for the proteins? So that means if a protein has to enter into the matrix, it has to contain a positively charged side, because it has to enter into the membrane and go, if it has to really enter into the matrix. All the proteins that we found until today, let's say, are the proteins that have always a positive charge domain to enter into the matrix, because this is where it's negatively charged. So this is a very characteristic of the proteins that are entering into the mitochondria. So I don't want to complicate here things, but you can just imagine different destination for the proteins. It can be the matrix, inner membrane, intermembrane space, inner membrane, or just inner membrane. So how do these proteins look like? So this is just a linear, very simplified structure of a protein, where it has an N terminal and a C terminal. And whenever you have a protein that has to go into the matrix or into the intermembrane, they always have to first use a gate, right? Because as I said, inner membrane is very impermeable. So you always need these specific gates to enter. And the gate that they use is a Tom complex and Tim complex, it's called. And these proteins have also this positively charged as the N terminal. And this is the site of the protein that is going in the matrix, and the rest of the site can go in the matrix, stay in the inner membrane, or whatever. There are always different combinations that are happening. So how this import is happening? Now, this is a very nice structure presented in a recent paper, which I wanted to share with you. So here we have two complexes. So there is a Tom complex and Tim complex. So just to simplify it, I wanna guide you through the slide. And you see in number 1, A1, you have there is a protein that has a positive charge at the site. It goes via the Tom complex. And then if it has to enter into the matrix, which is this part, it has to pass through Tom complex and then also has to pass through Tim complex. And it slowly slides into this complex, then goes into the Tim complex, and then enters into the matrix. Once it is in the matrix, this positive tail that is basically cleaved off, so that the protein does not have this positive charge unnecessarily. And this is basically chopped off by the proteases and then delivered to wherever it has to be delivered. Another way of translocating this protein is if the protein has to end up into the membrane. So what happens if a protein has to end up in the membrane? Then the protein comes again via the Tom complex. And then there are these small little machineries that are present in the intermembrane space, which we don't need for the part A, which we only need here. And here what happens is that the protein enters into these small Tim components and then enters into this last Tim23 complex into the inner membrane and is then transferred into the inner membrane. And that's how the hydrophobic proteins are then entering into the inner membrane of the mitochondria. So this was just a brief overview about how the proteins are transported. And my last slide is, of course, basically talking about mitochondrial disease and why mitochondrial disease are very, very important. It's because when there are mitochondrial disease, most of the time, if there is any mutation into the DNA of mitochondria, most of the time the result is just death. So there is still no way that we can actually rescue people from dying if they have mutations into the mitochondrial DNA. So that is, let's say, the worst that we don't want at all. And therefore, these diseases are also very rare. So the mutations into the mitochondria are very, very rare. If there are people that are affected by mitochondrial disease, most of the times they don't make up more than 5 years or 7 years in their life. So most of the times they would really die at the age of 5 and 7, which is very, very sad. And also their life, these 5 years of life that they live is not actually the life that you would wanna imagine for a kid. They are not able to walk properly. Most of the time they cannot see properly. They have problems in the limbs or the locomotor activity. The whole process of walking is basically disturbed. So this is the disease that are coming directly from the DNA of the mitochondria. But then there are diseases that are coming from the metabolic dysfunction of the mitochondria. So when the electron transport chain is not functioning, when the metabolism is messed up, or when the communication of the mitochondria with other organelles is affected, or when there is the dynamics of mitochondria that is disturbed. And these diseases can then lead to stroke, cardiomyopathy, mitochondrial disease, obesity, diabetes, and any disease that you can imagine. It can happen with the mitochondrial dysfunction. And I have to say here that mitochondria is the organelle where I think the most disease are associated with. So if you talk especially about heart, brain, and muscle, most of the disease are going through mitochondrial dysfunction. So if you take the heart of an animal that is sick, that has a cardiomyopathy, and you open this heart up and you check the function of the mitochondria there, it for sure is always affected. So I work a bit with these animal models, and until today I haven't seen an animal that has a cardiomyopathy, so a problem in the heart, and don't have a problem in the mitochondria. So it has been always like this, that whenever you open up the heart of a sick animal, you would always find mitochondria affected there, especially in the organs, like, as I always have repeated many times, the heart, brain, and the skeletal muscle. And with this, I let you ask questions, and I would like to thank you very much for listening patiently to this an hour talk. And you have been a wonderful audience. I didn't listen any disturbance or anything, so that was fantastic. And I'm very much open for questions. Thank you.
Okay. So this is Dr. Nicholson from National Engineering College. So thanks, Dr. Soni. Now it is open for discussion. So listeners, you can ask the questions. First, Prabha Priya has a couple of questions. So just unmute yourself and ask the question. Prabha Priya.
Dr. Soni?
Yes?
You may see questions at chat box.
Okay. So from Prabha Priya, I get the first question. Any research ongoing about injecting of father mitochondria into embryo? If it is so, what are the advantages and disadvantages? That's an amazing question. Actually, there is a lot of research going on in vitro fertilization, where basically people or scientists are trying to take the trying to create the baby, let's say, from basically 3 different donors, so 2 moms and 1 father. So far the father's mitochondria injecting into the cell has not been possible, because these organelles are so tiny. I cannot imagine how would you take it from the father's cell and then put it into the egg. But what people can do actually, or what has been done, there are very recent papers in Nature and Science where they talk about taking an egg, or not the whole egg, but just the cytoplasm of a woman that is healthy, and they are putting the nucleus, that is the mix between the mother and the father, and then you can put it into the cell and inject it back into the woman that wants to give birth. Because there the problem has been always the ethical question, because sometimes people don't want the kid to have a DNA from different parents. So if parents want to have their DNA into their kid, they would want the nuclear DNA at least to be from mother and father. So what they do is they just take this nucleus and put it into an egg from another mother that is more healthy, and then inject back this embryo into the mother's body. So the advantage is that it is possible. The disadvantage, so far we don't know. The survival rate of these kind of experiments is very, very low, because it's happening all in vitro and you have to do it all in vitro. The disadvantage can be mothers can say that the babies don't have DNA from them, but I think that is okay. So far, no really a big disadvantage. I hope that answers your question. Another question. Prabha Priya again, excessive reactive oxygen species leads to cancer because lipid peroxidation mechanism takes away a radical mechanism. What will happen when excess radicals produced? So as I said, so when excess radicals are produced, because these are very reactive species, they start reacting randomly to several proteins and several lipids. If this is directly causing the cancer, that is very hard to say and hard to comment actually, because cancer is a very complicated disease which actually starts with the defense mechanism that so every cell basically has to die if it is not working properly, right? What is happening in the cancer is that the cell is not dying. So the mechanism for the body to sense that this cell is dysfunctional is basically compromised. And this is the reason that you have the cancer, you have multiplication of these cells unnecessary. So this is what we know about cancer. That's the general information. How ROS specifically affects into cancer is not known yet. ROS is more connected to the heart disease, so cardiomyopathies, and neurodegenerative disease, so for Alzheimer's, Parkinson's, and all. And the damage that ROS is doing is basically making your proteins sick. So a protein is more oxidized, would not be able to function, making your DNA sick. If mitochondrial DNA is damaged, then the DNA is not able to encode for these very important 13 proteins that are there in the mitochondria. So that's how ROS is damaging. More questions? Mostly those oxygens can be neutralized by superoxide dismutase. Tamila Rasan. Yes, so as I said, that mitochondria is not something that just takes the so mitochondria is not something that you can like damage so easily. So they have a very, very strong defense mechanism, of course, and it happens that this superoxide dismutase is actually neutralizing the ROS. That can absolutely happen, but in most of the cases, this defense mechanism can be compromised under the pathological functions. So it really depends at what level these ROS are generated. If these ROS are generated as a side effect that your electron transport chain is not working, then of course for the dismutase like SOD is very hard to take over all the work. But there have been now clinical trials using something similar strategy. So using these proteins like SOD and others to basically kill the reactive oxygen species and see if there is an increase in the lifespan. And these experiments have been successful in the animals, but the clinical trials have failed massively, I have to say, and that's a huge loss for the community. So these trials have failed. Even if you can stop the reactive oxygen species, you cannot increase the lifespan of a human being. So human beings are not living more. Basically, this is not happening. So that means that we need a different strategy. And I think that the different strategy, what I learned in my PhD, can be that actually you can specifically target a specific source of ROS and specifically inhibit that source of ROS. Because as I mentioned, that reactive oxygen species are also very important for the signaling purpose. So if you are blocking all the ROS, that is not good. And we don't see the protective mechanism. So I think what we need to do is we need to block the ROS specifically under pathological conditions, but a specific source and not like in general any ROS. The quality control mechanism of the mitochondria is new to know by this lecture. This is, sorry, that was not a question. Okay. How to prevent mitochondrial disease from TA sugar? So that's a very cute question. How to prevent mitochondrial disease? If I would know, I would be a billionaire today. So, okay, how to prevent mitochondrial disease? I think it's very important to understand the pathogenesis first. So very important to understand the molecular mechanism by which the disease is happening. And I think once we can know the molecular mechanisms that are causing the mitochondrial disease, we can specifically block the pathway, the particular pathway. And this has been now ongoing very, very rapidly. Most of the pharmaceutical companies in Germany are actually synthesizing the compounds that can block a particular enzyme that is like a rate limiting enzyme or something in a particular pathway that is maybe enhanced in diabetes, or that is enhanced in cancer, or that is enhanced in any other disease. And I think that is the only way we can go forward. The side effect of this kind of approach is that the delivery of these compounds into the body. So how would you do that? Because you need to imagine that this compound has to not only enter into your body, but also has to enter into the cell and into the mitochondria. So if it's a specific pathway for mitochondria, for these compounds to enter into the mitochondria based on the electrochemical gradient that this organelle establishes is very, very tricky. And there is a very good lab in Cambridge, where a very good scientist, Mike Murphy, is working in developing the compounds that can be targeted to the mitochondria. And he has been generating several of compounds where he has tested on the heart disease. And there are some compounds that have worked, but again into the animals so far. And I hope that he pushes the compounds into the clinical trials, and then only we can know something. Ma'am, could the prolonged medication, say diabetics, hypertension, or others, induce mitochondrial disease? So before, people were thinking that mitochondrial dysfunction is the cause, is the consequence of the disease. So you have a problem in the heart, your heart is not working, and as a consequence, your mitochondria fails. But now, as I said, more and more research is progressing, and we are realizing that it's rather the consequence of the disease. So it's happening more and more that mitochondrial dysfunction is coming up as the cause of the disease. So I think that when you have diabetes or hyper blood pressure, actually your mitochondria is already most likely already damaged. In my PhD, I work with animal model with diabetic cardiomyopathy, and there I have assessed several animal models which had heart problems. And any heart that I opened or I looked into the mitochondria, all the mitochondria were always dysfunctional. So I guess what you mean by mitochondrial disease is that mitochondria is dysfunctional, and this is, I think, already there when these diseases are there. So any more questions? Is there any age factor like from this age to that age we can get this disease? Rashika, thank you for the question. Any age factor? Yes, absolutely. So there is not a specific defined age that it happens from this age on or something, but most of the time people often above 60 years old, or from, in India, let's say from 50 years old, in these people with the aging, it can happen that all the function starts to collapse a bit. So how it happens is that the first thing that is usually mitochondria is giving up is the quality control system. So of course, everything can work well as long as your defense system is really, really strong. So if something is not working, as long as your defense system is strong, if you can get rid of the garbage, and if you can add fuel to the machinery, it will work, right? But the problem is, if the quality control system is not good, then even if your engine is working fine, this garbage will start to interfere at some point. And I think that is one of the biggest problems. So with age, we have realized that the quality control system of the mitochondria starts to decline in general, also of the cell, and that creates a lot of problem. But what exactly are the patterns and how exactly it happens, or why with age it happens, this no one knows. Whether weakness in old people is due to the energy collapse in the later ER stress. So weakness in the old people, this is actually because if you so you can do a very nice experiment. This is really possible. You can take out the mitochondria from a young animal, which is 5 weeks old, 6 weeks old, and you can take out a mitochondria from old animal. When I say animal, I mean mice. Our model system is mice usually. So from these mice, when you take out the mitochondria from the old mice, which are 2 years old or 2.5 years old, which corresponds to roughly, I think, 60 years of human. So there, if you isolate this mitochondria and you look at the electron transport chain function, that you can look, because you can measure the oxygen consumed by these organelles. Okay, because of course at the end what is happening is oxygen is consuming, accepting all the electrons. So what is happening is that if you isolate these mitochondria from the young and the old mice, you can really see that a young mitochondria performs way much better than the old mitochondria. So I think this I haven't done in humans yet, but I would imagine that if you also take out mitochondria from humans that are young and humans that are old, I bet that this would be the case, that mitochondria would not be able to consume the oxygen that a young mitochondria is consuming. So this tells us, this can only tell us that a mitochondria is basically functioning less. So as a result, you have less energy produced, and as a result, you are not so active, right? And that's why you feel weak. One way to go around this could be if you exercise and you train your muscles and you train your body in a way that it keeps producing more and more energy. Then it can be one of the way that your mitochondria still remains healthy, because it is adapted to this huge energy consumption and it is producing more and more energy. Is the same like how we can take an example within our body between heart and the liver. So heart is the one that is working more. Therefore, the mitochondria in the heart is adapted to produce more and more ATP, whereas the mitochondria in the liver is working less, but still working always, but less as comparison to the heart. And there, mitochondria is consuming less oxygen. So maybe when the time comes, and if you put these two mitochondria on a race, heart mitochondria would perform better than the liver mitochondria. But if you train the liver to also work so much like heart, I'm sure the liver mitochondria eventually would also go up there. So that's why where the physical activity becomes very important in our life, that we should train our muscles and we should train our mitochondria to produce more and more energy so that you don't feel weak. And that was from S. Sudarshan. Yeah. So I don't see any, oh no, there are. Thanks. Okay. So I don't see any more questions here. I saw some comments where you ask for the presentation. I can send it to Ramachandran, and maybe he can pass it to you. And if you guys have any question or something, he also has my email address. Please feel free to write to me if you have any questions or if you want to know something more about mitochondria. So I think that would be it from my side.
Okay, thank you, Dr. Soni. That was a powerful presentation about the powers of the cell. And also, you have explained in a nice manner how it communicates with the organelles inside the cell. So thanks a lot. And I would like to thank Dr. Ramachandran for arranging such a wonderful webinar series from Monday to Friday. So this is the 5th day. So a big thanks to Dr. Ramachandran. And I would like to thank our management, our head of the department, principal, and all the professors of National Engineering College. And finally, I would like to thank all the participants throughout this 5 days. A very big thanks to all the participants. So once again, thank you all. Thanks a lot. Thanks, Dr. Soni.
Thank you so much.
Thank you, Dr. Soni.
My pleasure.
Thank you very much. And now I would like to thank our management, director sir, principal, and our head of the department, Professor Dr. M.A. Neelakantan, for giving me opportunity to organize this 5 days international webinar for engineering and science students. And thank you for all your participants for your passion, and continuously you attend this webinar on 5 days. So we will send some certificate link to your email ID. So after filling the certificate link, you will get the certificate. So please carefully enter your email ID. Thank you. Thank you, Dr. Soni.
Thank you. Bye.
Bye bye.
Bye.
Okay.